Welcome to Season 2 Purdue University College of Sciences Superheroes of Science podcast. I'm Stephen.
And I'm Sarah. We will be discussing anything and everything related to science. If you have a science question, tweet it to us at Purdue SOS, and we will try and find someone to answer it for you. Welcome to Purdue University Superheroes of Science. We're here today with Betsy Parkinson, Assistant Professor of Chemistry. Welcome, Betsy.
Thank you. I'm glad to be here with y'all today.
Yeah.
All right. What'd she say? What were you again?
I'm
Professor of what?
Chemistry and molecular pharmacology. So.
Whoa. I'm not sure where to start then. How does chemistry and pharma what?
Medicinal chemistry and molecular pharmacology.
Okay.
How do those go together?
Yeah, so one of the things that I'm really interested in is actually finding new antibiotics. And so a lot of the antibiotics that we use are chemicals. So that's kind of how the chemistry gets in there. And the medicinal chemistry and molecular pharmacology, that mouthful, is also an aspect of developing drugs. So it's actually a part of the department of the College of Pharmacy. And so it's housed within that.
Cool. So you're interested in antibiotics, and what projects do you have going right now?
Yeah, so the major project that I'm really interested in is actually trying to find new antibiotics from the soil. So kind of the overall theme of my lab is from microbes to medicine. So we actually go into the soil. So it's really neat. People usually think of dirt as just kind of dirt. You don't really think about what's in it. But if you actually pick up a handful of dirt, over half of that is actually some sort of microorganism. So a bacteria or a fungus or something along those lines. And the dirt is actually this really complicated environment where things are competing constantly. It's like chemical warfare. So we can actually find antibiotics in the soil because they're trying to actually kill off some of their competitors. So I study a specific kind of bacteria called Streptomyces, which are really common soil bacteria.
So the bacteria that's naturally in the soil is creating an antibiotic?
Exactly.
How and why?
No, it's kind of bizarre. You think, that's strange. Why would they want to do that? So you asked how, so I'll answer the how first and then get to the why. So the way they actually do these is, so all organisms have DNA, right? So DNA, so there are genes within that DNA that encode for proteins. So kind of central dogma, DNA, RNA to protein.
Okay.
So there are kind of genes that are all bunched together, and we call these biosynthetic gene clusters because they're all clustered together. And these genes actually encode for proteins that can make the antibiotics. So they are enzymes, so they catalyze reactions, and they take molecules that are typically in the bacteria, so kind of standard metabolism, things that you normally find in most cells, and can convert them into antibiotics using these enzymes.
Wow. And when you say antibiotics, do you mean like the antibiotics that I give my children when they're sick and that I take when I'm ill? And
so approximately 70% of antibiotics are quote-unquote natural products. So what a natural product is that it is a molecule that came from a living organism. So it could be a plant, it could be a bacteria, it could be a fungus. Of those 70%, the majority actually come from soil-dwelling bacteria. So vancomycin is one of the ones that's used as a last resort. So if you have a resistant infection, that's used. That's from a Streptomyces. Daptomycin is another one that comes from a Streptomyces.
Wow.
So how, I'm still trying to get my head around this. I mean, how do you get something from a bacteria into something into a syringe?
It's a long process. So we don't go out to the dirt, obviously, and pick up a bacteria and feed it to you. That's not how this works. Essentially, it's many, many years of research. So the first step is figuring out what that molecule is. So what we'll do is we'll actually go out, get a bacteria we'll actually get a scoopful of dirt.
Really?
We'll isolate bacteria from it.
Okay.
I've done it I'm a farmer's daughter, so I grew up in Mississippi on a cotton farm. And I've gotten dirt from my home farm and isolated Streptomyces from it.
Wow.
So, yeah, it's very easy, actually, to do.
It's like a fairly common bacteria then that we're talking about?
It's a very common bacteria. So it's isolated all over the world, so you could find it in your own backyard, but it's also found in the desert, in the Antarctic. At the bottom of the ocean, there's some in the kind of dirt at the bottom of the ocean as well. Some live on sponges in the ocean, and some actually live on ants. So there's a person at Harvard who actually studies the ones that live with ants, and what's actually kind of cool is they produce a molecule that defends the ants from a fungus. So they're like a symbiont.
Oh,
Wow.
Yeah.
okay, that's pretty cool. Now, does sometimes, I mean, is it always a beneficial thing that they're producing?
So they produce these molecules to benefit themselves. So they make some molecules that wouldn't necessarily be quote unquote beneficial for humans.
Okay.
But they are generally a very safe organism. They don't generally produce a lot of toxins or anything like that. However, they also produce other things. So like if you walk outside right after it rains, you know, when you take a deep breath in and it smells really
I love
good,
that smell, yes.
Yeah, I do too.
that's actually a molecule made by these bacteria. It's called geosmin. And so the rain kind of disrupts the dirt and causes that molecule to go up into the air, and that's why you can smell it.
Oh my goodness.
Oh.
Ah.
And so when people like compost and stuff like that, is there a higher percentage or more of this type of bacteria, or is that a different type of bacteria when we talk about?
So there are lots of different bacteria that are within compost, and so these specific bacteria do not necessarily grow on decaying material.
Okay.
They can, but there are lots of other bacteria that also do that too. So.
Okay.
Yeah.
And you might have just said it, I don't remember. I'm still wrapping my head around the whole, how we're getting medicines from soil, which I knew we did, but I'm like, wow.
I didn't know. That is really awesome.
It is,
Yeah.
but they're producing it for a defense thing.
Right.
So it's like they're secreting this to defend their turf.
Exactly.
From other bacteria?
So, from other bacteria and fungus. So, when they're actually in the dirt, there are lots and lots of organisms that are competing for nutrients. And so, it's not dissimilar to like two lions in Africa competing for the same elk. They are competing for the same nutrients, and they want to have an advantage. Now, some bacteria are modal, so they're able to move, but a lot of them are not. A lot of them cannot necessarily control where they're going. So they're stuck on their little piece of dirt, and they have to survive. And so in order to kind of avoid some of the competition, they'll make these molecules that can kill off their competitors to try and steal their food, essentially.
Okay.
And how do we make the jump from these things that they're producing then to medicine?
Yeah, so the first step of it is structure elucidation. So that's a fancy term for figuring out what atoms are in it and how they're connected to one another. So we do a lot of experiments involved with that and figuring out what connectivities they have. So how many carbons in it, how many nitrogens in it, how many oxygens, and what patterns do they form. And so once we have that structure, the next step is really to kind of test it further. So test kind of the types of bacteria it's good against, and also whether or not it's toxic to humans. Because some of the molecules that these bacteria produce, they weren't producing them to make drugs for us. They were producing them for them. So some of them will be toxic. So the way that we do that in my lab is we actually grow mammalian cells in culture. And so we will test kind of the molecules that we get, both on bacteria as well as on these mammalian cells. And if they kill bacteria, but they don't kill mammalian cells, that's a good sign that likely they will be safe.
Okay.
And so once we kind of have that, the other aspect of this is making sure they have good properties. So ability to make a good drug. So again, these weren't designed to be swallowed by us, go into our digestive tract and absorbed into us. So some of them are not very They don't dissolve very well in water and things like that. So sometimes we'll actually, so I'm also an organic chemist, so one of the things we sometimes do is either modify what we've gotten from the dirt, so we'll use chemistry to make small changes that hopefully don't change the activity, but will change some of their solubility and other properties.
Wow.
Or we'll take it from the bottom up and we'll do what's called a total synthesis. And so we'll start with very small building blocks and build up from the bottom to create a molecule that's very similar to that molecule we got from the bacteria, but might be a better medicine for.
Okay.
Okay.
I've read recently in the news that just antibiotic resistant bacteria or superbugs are kind of here, and
Yes.
and I just remember thinking, so will there come a time where we won't be able to take antibiotics anymore? So
Yeah. So
is there
that's,
anything you have to?
so that's a very scary thought, and it is something that's very prevalent in the news. One of the major things that we want to do is keep doing research. So there's a desperate need for more research to find new antibiotics, because the fact of the matter is bacteria have a around 20 to 30 minute life cycle, so they divide very often, so they can evolve much faster than we can to develop this resistance. So we're a little bit in an arms race where they have a lot of jump on us because of that. However, at the same time, one of the things we can do to start thinking about combating that, so research to find new things. Another is good stewardship. So if you have a cold and your doctor tells you it's viral, don't ask for antibiotics, because that's one way that we kind of make our antibiotics worse, because that's more likely to cause resistance to develop within you. Another thing is actually one of the things that we're trying to do is more co-treatments. So treat with more than one antibiotic at a time. So different antibiotics kill bacteria in different ways. So some of them might damage DNA, others of them might, for example, attack their cell membrane or their cell wall. And if you attack in one way, it's very easy for the bacteria to overcome it. But if you attack in two different ways, it's much harder. So utilizing combination treatments is another really good thing.
Yeah, almost kind of tricks them maybe, and they can't.
Yeah, they don't know which way to change.
Yeah.
So,
Wow.
Yeah.
That's really interesting.
Yeah.
So now your focus is on looking at the bacteria that's in the soils and then seeing what type of antibiotics come from that.
Exactly.
Now, do you overlap into? I'm thinking like these antibiotics. I mean, obviously, well, not obvious. I'm guessing that they target certain types of cells and certain things. And so, is the things you're looking for? Is some of it going to target like a cancer cell versus a flu bug versus whatever?
So you're exactly right. There
Okay.
are many molecules.
Write that down.
So there are many different types of molecules coming from these bacteria that do different things. So some of them are being made to kill other bacteria.
Okay.
And that kind of makes sense. Some of them are being made to kill fungus. So they're fungus in the soil as well. And so antibiotics has been my focus, not antifungals. I just have a history with that.
And an antibiotic is one that's killing the bacteria itself?
Exactly.
Okay.
Exactly. Antibiotic kills bacteria.
So wait, I'll let you finish. I'm
Okay.
throwing questions on
No,
questions.
you're
You're not getting to
good.
finish. I'm so sorry.
No, you're awesome. So we also look for anti-cancer molecules as well in there. And so those, it's a little bit harder to explain why they make them, because the competition in the soil argument makes perfect sense for bacteria and fungus.
Yeah.
But it doesn't really make sense for cancer. And there isn't a great answer to that, actually. That's something we don't really understand still, and are still thinking about why would they make a molecule that could kill cancer. There's some level of cancer where eukaryotes and fungi, and there's some overlap there. There's also some that attack plants, and we might share some targets with plants. But there's some that we just don't know. For example, one of the cholesterol-lowering drugs comes from Streptomyces. I have no idea why they would want to make it a cholesterol-lowering drug. Doesn't make any sense, but they do. So.
Wow.
So could you explain what exactly is the difference then between, like, as you mentioned, a cold viral. So what's the difference between a virus and a bacteria? And I guess and a fungus?
That's a good question, yeah.
Can you explain? Because we hear, oh yeah, colds are viral, antibiotics doesn't work for that. Suck it up, suck it up, wash your hands, get over it. But if it's something more serious, then you can take medicine to help combat that. So
Right.
what's the difference between bacteria and virus?
Yeah.
And then throw in the whole
Fungus.
thing.
So I'm going to start with bacteria versus fungal.
Okay.
So bacteria are prokaryotes, fungi are eukaryotes. So just kind of this initial difference in kind of their cell structure. So your eukaryotes are going to have your cell, but they're going to have different organelles, almost like mini organs within them. And they have different regulatory systems. And so the way they kind of live is different. Some of the signals they have that are different. So for example, the way they make proteins, they both use the ribosome to make proteins, but they're slightly different. So a molecule that targets the ribosome in a bacteria is different from one that targets it in, say, us. So we are actually able to take some protein synthesis inhibitors that only target the ribosome in a bacteria and not in a fungus or not in a human. And that's why we don't have terrible toxicity. So we don't have bad side effects from it. As far as bacteria versus viruses, so bacteria are able to live all by themselves generally. So they're able to divide. They don't have to generally infect another host in order to survive. Viruses depend on their ability to infect another host. So they kind of are not really, they're not really fully alive. There's a little bit of controversy about this, but typically viruses have some sort of outer capsid, some sort of DNA or RNA, and then some ways to a very few genes. They have a lot fewer genes than, say, bacteria that can kind of replicate itself. So they're different. They're just completely different. So the drugs that you would take for example, a virus, so some of your anti-HIV drugs are not going to work on a bacteria, and vice versa. The things you take for bacteria
infection
are not
strep throat or something.
They're not going to work on viruses just because they're different.
Okay.
I don't know if that makes sense.
Well, yeah, it does.
Yeah.
It does.
Okay.
And I was like, I'm curious. Yeah, it's sorry, half these things are just me, my lack of knowledge asking questions.
No, that's great.
But I'm going to put you on the spot. If I have to cut this out, I will.
Okay.
Feel free to tell me, no,
Sure.
we're going to cut this out.
Okay.
But, all right, so this, I keep hearing now about the news. The biggest thing on the news, other than the lame politics stuff, is the coronavirus.
Sure, yes.
And that's a virus?
It is a virus.
And not a bacteria?
And not a bacteria, yes.
Okay.
So they are not going to be able to give antibiotics for this virus. There are people who are working on antivirals. So antivirals are things that kill viruses. And so there are already people that are trying to work and find things that can kind of stop the coronavirus. But no, you cannot take an antibiotic to stop the coronavirus.
Why does it, why am I hearing so much about that virus versus the whatever we got our flu shots for? Or is that a virus? No, it's not a virus.
Yes.
Okay, it is. Okay.
Yes.
But why am I hearing so much more about that all of a sudden? It seemed like a week ago this just popped up, and now it's like they have entire cities quarantined and stuff. I'm like, whoa.
So there are a couple of different aspects to this. The first is it's new. So the flu's been around for years and years and years. So we do hear about it every season. But because it's new, it's something that we don't know a lot about. And so that can be kind of scary. It's relatively highly infective. So there are different scales for how infective things are, and it is slightly more infective than the flu in the sense that it's more easily spread. So it's spread if someone coughs and you breathe it in, that's the way it spreads. I think the reason that there's so much news about it is why we know is because we know so little about it.
Okay.
So at least I'm not an expert on coronavirus
Yeah,
or
yeah.
viruses in general.
And that's
Full
what I said.
disclosure.
If I put you in the spot, I apologize.
No,
It
you're
was just
fine.
some courage.
I-
I'm a
yeah.
scientist that studies
No.
things. Don't ask.
Yeah, I feel like I should send you over to the virologist for this. But so my understanding, at least right now, is that we're not really sure whether it spreads before people are symptomatic or not. So that's a hard issue because many spread pre-symptomatic.
Mm-hmm.
So if you get on a plane and you feel fine, and the next day you get sick, well, they might have
yeah.
it might have actually been spreading before you even knew it.
Mm-hmm.
And so that's one concern. Another concern is the death toll and how many people are dying of this. And right now, we don't have a good sense of that, whether or not it's really going to be bad or not.
Mm-hmm.
So for some comparison, a couple of years ago, one of the closely related viruses, SARS, had a 10% death rate. Right now, they're guesstimating that this one's around 3. I don't know the flu off the top of my head, but I would guess that it's probably pretty similar. So I think that if you're in China, it is a serious concern and you need to be worried about it.
Mm-hmm.
Here in the United States, I would be more worried about the flu. Go get your flu shot, make sure you got that.
Right.
Yeah. Unless you have traveled to China right now,
Mm-hmm.
I think there are 5 people in the States with it and all of them
that's
came
what I
from
saw.
China.
Yeah.
So
Yeah.
Well, if we switch back over, can we switch back over to bacteria?
Well, yeah, because that's where I was going to
Yes.
go back to. I was
Okay,
going to go
great.
back to
yes,
that
that make me a little more
Yeah, right.
comfortable.
too. Leave it to
Thanks
me.
for
I'll leave you,
entertaining.
make people uncomfortable.
Yeah.
No, you're good.
That's what I do. That's why I'm here.
Well, I just wanted to go back to when did you first get interested in looking at the soil when did you first learn about that and decide you could get samples from soil and isolate bacteria and things?
So it was when I was in graduate school, actually. Okay. So I went to graduate school because I wanted to become an organic chemist to develop new medicines. Okay. So I did my undergraduate research at St. Jude Children's Hospital and got really interested in specific cancer treatments because I volunteered with the kids and saw they have a lot of bad side effects from a lot of the medicines that they use and wanted to kind of help develop some more specific, selective therapeutics.
Mm-hmm.
And one of the things that I did in my graduate lab was actually chemically synthesize one of these natural products that has this selective anti-cancer activity.
Mm-hmm.
And when I started, I didn't know where this molecule came from, but as I went through my PhD, I learned more and more about how it comes from one of these soil-dwelling organisms. And the other aspect of this is building molecules from the bottom up is very cool, but very challenging and very time-consuming.
As you say, how much time are we talking just for one molecule if you're going to start it?
It depends, obviously, on the complexity of the molecule.
Mm-hmm.
So for me, so the first aspect of this is developing the route, so figuring out how you're going to do it. And I will say that the molecule that I worked with another graduate student on this, and prior to us working on it, he really developed the original synthesis, and it took him about 3 years to develop the synthesis.
Wow.
Once we had the synthesis in hand, it usually takes a couple of weeks to actually synthesize it, but that's a relatively simple one. There are others that I know people who spent their whole PhD, 5 years, trying to build a molecule from the bottom up. Whereas these bacteria can do it in a couple days, not even.
Wow.
So they're so much better at it than us. Don't get me wrong, organic chemists are great, and I have tons of respect for them. So please do not misinterpret that, but just the fact that they are so efficient at making these just is fascinating.
Wow.
And that's why I decided to go into them.
That's really,
All right, so how much? All right, so we have some slides, right?
Yeah.
And if you're just audio, I apologize. We'll kind of describe the best we can. All right, and so this big old blob thing.
Yeah. So what we have in this picture are 2 different, we call them colonies of Streptomyces. So bacteria, they will start as a single cell, and that single cell will divide and divide and divide until it's big enough to be visible, and that's called a colony. And what's kind of cool about these two is if you look, so the top one there, it actually isn't perfectly round. They typically are perfectly round. And so what we can see is the bottom one is also growing, but it seems to be growing pretty well, perfectly round. Mm-hmm. So that suggests that it is making a molecule that potentially is able to kill the other one, or at least prevent its growth. The other thing I love about this image is you see the red and how it's much brighter at the bottom.
When it's close to the other one, it's really bright.
So that's it responding to that. So the bottom bacteria colony is making something, and then this bacteria is feeling it, and it's upregulating the production of this red molecule closer to this molecule.
And so what would my scale be? Is this something that you'd have to use like a microscope to see?
So no. So I would say that top colony is probably 0.5 centimeter across.
Okay.
So these are pretty big. I've let these grow for a while.
And approximately how many cells?
Oh, millions.
Okay.
Millions.
Yeah. Okay. Right.
Hundreds of thousands at least, yeah, a lot.
Okay. And so you would, all right, so if one can't be circular, then it tells you one adjacent to it is preventing that growth. Now, how do you extract what they're emitting to prevent it?
Yeah, so how do we actually get at those molecules? How do we get molecules in tubes? That's a great question. So the way that we grow these is we actually grow these on petri dishes on solid agar. And so the agar's kind of like Jell-O. It's kind of that same consistency. And so we'll grow them on this agar for a while. And then what we actually do is we then freeze it.
Oh.
And then we thaw it. And when you do this freeze-thaw cycle, it actually kind of breaks down the agar a little bit, and you can actually squeeze it. So we actually squeeze the agar, and the molecules that they produce, they spit out. So the bacteria make the molecules inside their cells, but they spit them out because they're using them for defense. And so when we squeeze that agar, we get all the molecules out. We then also do something called an extraction on the agar. So we take that agar and we will incubate it with different organic solvents. So if you've ever used acetone as nail polish remover, acetone is an organic solvent. The alternative, the non-acetone nail polish removers, are ethyl acetate, another organic solvent. And that's actually the one we use in the lab a lot, is ethyl acetate.
Oh.
Also methanol sometimes. But
I've heard of that.
Yeah.
All right.
The next one.
I'll jump through to the next and let you tell us.
There you go. Okay.
all right.
That doesn't like my fingers.
Yeah, you must be too dry.
Yeah.
It's winter. You must be too dry.
Yeah, so for this one, the one kind of on the upper left is the same type of bacteria that was on the bottom on the slide before. So if you kind of squint at it, you can, and if you go back real quick, can we go back?
Yes.
You see how it has a similar morphology.
It reminds me of bouquet.
A little bit, yeah. So we can go forward. But so morphology is one way that we tell Streptomyces apart. And it's kind of the old school way, but it's one of the ways you tell which strain is which. It's just by what it looks like. The one in the bottom right corner, I really like this one because you can see a little bit more of its three-dimensional structure. So you can start to see that there are actually areas that are kind of growing up. And so Streptomyces are kind of strange in that they grow in they grow 3D almost. And that's part of their life cycle, is that growing 3D will actually lead to their spore formation, which allows them to continue to replicate.
How are these different from the orange that I left on the counter too long that it was growing in 3D too?
Yeah, so probably the orange you saw growing on your counter, had fungus on it. Was it green and kind of furry looking?
Yes.
It probably was fungus, not bacteria. It might have been bacteria, but a lot of times that is
Okay.
going to be fungus
Okay.
for what's growing.
Which requires a symbiotic relationship, a fungus does?
Not necessarily.
No?
No, not
Okay.
necessarily.
Oops. I'm thinking of lichen.
Viruses.
Oh.
Lichen. Yeah.
Sorry.
Yeah, no, you're
I went totally into the, we'll go to the next slide now.
Yeah.
Ooh, this is a new one.
I like this one a lot too. So the one on the left, what I think is really cool is that most of the way around you can see that kind of white ring.
Yeah.
But
Mhm.
right next to that other just colony of bacteria, you no longer see that. And so it's somehow inhibiting the production of something. So we don't understand this, but this is kind of an interesting phenomenon that we saw. Also, the one on the left looks kind of fuzzy,
Mhm.
right? Those are the spores. So this one's already sporulated, and the fuzzy look is spores.
And the spores, and that's it would be like emitted and producing more.
Exactly. So spores are very hardy. So if it got into a bad environment, the mycelia, which is the other part, would die, but the spores are heat resistant. They're resistant to vacuum, cold, pretty much everything, and they'll
Wow.
just survive. And when conditions are right again, they'll
Start growing.
Oh. Nature has a way.
Ooh.
So this one I find fun. So a lot of these bacteria, in addition to making very interesting antibiotics, also make really cool colors. So you see kind of the bluish color is actually a molecule called actinirodin. And actinirodin is actually a pH indicator. So it's
Oh.
blue at certain pHs and will turn red at other pHs. So it's kind of I just think it's pretty, honestly.
It is.
And this is the same one, just kind of close up. But
These are pretty good size then, since I can see the side of the
Yeah.
petri dish
Petri
as
dish,
a
yeah.
comparison.
exactly.
And this was from your lab.
Yep, these are all grown in my lab.
Neat.
These are all pictures I took. So
That is neat.
I think the next one's my favorite. Nope, we're not to my favorite yet.
I like this one because it's like a little volcano almost. They almost
It
look
does
like,
look
which I love, but
I didn't know how to describe that. Okay, we'll go with that.
mountains, I don't know. And again, you're starting to see spores. So the white,
okay.
spurry stuff,
Yeah.
again, is the spores that they're forming.
Okay. When I first looked at this one on the screen, it looked like the one was trying to grow out of it.
Yeah,
Like you
exactly.
have an
It's right on the
escapee.
edge
Yeah.
of the petri dish.
Yeah.
Do you have, that's, I guess a question I should ask. Do you, when you're growing things like this,
Yeah.
is there a safety concern sometimes?
Not at all. So these are all what are called biosafety level 1 bacteria. So what that means is that they do not cause infections in humans. So they are not going to cause anyone to get sick. You've probably eaten some of them if you ate mud pies as a child,
Oh.
and you're just fine. So
Okay.
yeah,
Okay. Oh, there's the one.
Aw.
I like that one a lot too. So we don't know for sure what the pink color is, but a lot of them make a pink pigment that's actually used as part of their electron transport chain.
Okay.
So we think that might, because the conjugated systems oftentimes are colored, and those are also oftentimes used in energy transfer. So.
Oh, wow. And these are all things, you've grown all of these
Yeah.
just from soil samples.
Yep.
And if you're listening in on the podcast, this is a picture of some different petri dishes arranged in the shape of a heart with the inscription "Happy Valentine's Day" from the Parkinson lab and our microbes. So it's a cute picture. Check out YouTube.
Yeah, that's. And then when I saw this one, I thought of like the pictures I've seen of fat cells.
Yeah, a little bit. So what this is showing is how we streak out the bacteria. So we store bacteria actually as frozen stocks, so you can freeze them and leave them in your freezer, and they're completely fine, they're completely viable, and you take them out. And when you revive them, you physically streak them, so we actually just get a toothpick and get a little bit out of the freezer, essentially like scraping an ice cube with a toothpick,
Uh-huh.
and then just kind of streak it on the plate, and this is what you get.
Oh. Huh. Oh, that's the last one of those. We'll go back to our splash screen.
Oh, there we
We'll
go.
go back to us.
All right. Well, Betsy, we really appreciate you taking the time to tell us about your research today. So thank you for joining us.
Yeah, thank you for having me. It's been a lot of fun.
Yeah, I mean, I always knew soil was pretty nifty, but I guess I never realized that microbes, when they're battling for territory, and their battle is what we're extracting for our health.
Exactly. You got it right.
That's really neat.
That's crazy.
Yeah.
I like it. Thank you.
All right.
Thank you for listening to our podcast.
Be sure to subscribe, rate, and leave a review.
An outstanding
on
review.
iTunes or your preferred podcast player.
Tweet us your science questions.
At Purdue SOS.
Until next time, be super, and remember...
You are someone's hero.
Boiler up!
Hammer down.